Hello and welcome to the webinar. If you've just joined, I'm Jessie Kennedy. I'm with Dovetail Genomics, part of Cantata Bio's marketing team. And it's my pleasure to kick us off this morning or this afternoon or this evening, depending on where you are in the world. It's a pleasure to have you with us. And today we're going to talk about 3D genomics and why it matters. So let's get started here. Again, I'm Jessie and I'm part of the marketing group. And today our presenter will be Cory Padilla. He's a product manager with Cantata Bio and he's been with us for quite some time. Cory has been with us actually since 2017. He received his PhD from the Georgia Institute of Technology in cellular and molecular biology, and he holds a BS from UC Santa Cruz here locally. Cory joined Dovetail in 2017 as part of the research and development team, and now he leads product management. His focus is to act as a liaison between the company and the scientific community, as he's doing here today. In this role, he identifies new research area, he captures feedback on customer needs for integration into product development, and he works to educate the community on advances and new products from Dovetail. Cory spent the last couple of years driving development of informatic pipelines and bespoke data analysis on mRNA-based proximity ligation. And he'll be talking a bit more about that today. So today's going to cover how 3D genomics fits into the multi-omic toolbox. And he's going to cover ChIP-seq and ATAC-seq and how these dovetail into other types of analysis that you might already be doing. What information the 3D genomics uniquely unlocks, and why integrating 3D genomics provides valuable insights into your research and what you can do with it going forward. So pretty exciting. Today, we would love to hear from you. If you have any questions, please submit them in the Q&A box. Please don't use the chat window as we won't be monitoring that, but we will be looking at the Q&A window. And if you have any questions, you can drop those into the Q&A window as Cory is speaking. And then when he takes a break, he will address those questions. And then afterwards, please watch out for an email from me, Jessie, because I'll be sending out the recording as well as Cory's slides. We also wanted to let you all know that as a follow-up to this webinar and to all the webinars we do, we offer a deeper dive in more of the nitty-gritty or nuts and bolts of how these experiments are run. And that is going to be presented by our speaker, our senior customer success scientist and specialist, Miriam El-Kawand. And she'll be doing that next week on August 9th at 8 p.m. So if you're in the Asia Pacific time zones, that's better for you because that's going to be about 11 a.m. the following morning, or on August 15th at 8 a.m. Pacific, which is more amenable to the East Coast, US and Europe. She's going to cover how to design 3D experiments, what's involved in running an experiment in the lab, and how to navigate the data processing. So that'll be really valuable if you're just embarking on doing this kind of research. All right, without further ado, I'm going to hand it over to Cory, our illustrious speaker.
Thanks, Jessie.
Here we go. All right.
I am going to grab the screen. Actually, Jessie, if you could stop sharing.
Yeah. Let me do that.
Great. As Jessie mentioned, we're going to be talking about the 3D genome today and really why it matters and how you can integrate it to really help drive your research. So really moving to a more causal understanding of the system that you're working on. But I really wanted to start off with these sort of quotes from recent publications. We had these up as folks were joining in the webinar. I think it's really important to hear from folks that work outside of the company of Cantata and that deal with the Dovetail products on a day-to-day basis to hear what their viewpoints are. And I think there's a really powerful future ahead when it comes to 3D genomics when you think about the clinical relevancy of some of the features that can be captured in the 3D genome and the function of being able to link SNPs and causal variants to their target genes. It's a really big area of research moving forward, kind of illuminating that dark matter of the genome. Another thing to think about too is the 3D genome is an entirely new mechanistic view to a lot of people in their research programs, and it can really help, gain insight into new biomarkers or potentially new target areas to really focus in on, because it's a completely different dimension of data that most folks haven't been integrating, but are starting to. And then it's really powerful to integrate with other multi-omic tools like ATAC and ChIP-seq. And these types of multi-omic approaches can really help develop our understanding of cancer formation. And we actually have some really nice case studies around that later on in the talk. So about the talk, we're going to do a very brief intro to 3D genomics, kind of what it is, what are the features that you can capture, then go into the case studies, and then finally, how would you access the 3D genome? So understanding 3D genomics, what is 3D genomics and where does it really fit into our understanding of molecular biology? Well, a lot of the research that we do in biological science is really to understand the journey from genotype to phenotype. How do we go from this encoding of primary base information all the way up through phenotype, whether it's a normal phenotype or whether it's a disease phenotype? We really strive to understand, going from that molecular level all the way up to the phenotypic level. And we try to understand this by looking at multi-omic approaches, right? Whether it be the genome, that primary base structure, the 3D genome, which is what we're going to be talking about quite a bit today, the epigenome, the transcriptome, the proteome, all the way up to the metabolome, right? What is it that these proteins are actually generating in terms of helping give rise to that phenotype? And these multi-omic approaches, each one of these provides a very unique insight into the biological questions that we have. And so if we go through these stages, right, the genome gives us that primary base information, right? It really tells us, does a base switch from a G to C or T to A or, G to T, whatever. It really allows us to understand what the primary information is that's captured in the genome. The 3D genome allows us to map DNA structure inside of a cell or inside of a nucleus. And this is going to become really critical when thinking about the more upstream components like the epigenome, which is all about mapping factors like methylation, chromatin accessibility, histone modifications, things like that. And then thinking about all of those things in light of the transcriptome, what genes are actively turned on? What is trying to be converted into protein? And the proteome is assessing, right? Well, what proteins are actually in a system? What is their abundance? What's the diversity of those proteins? Things like that. And finally, the metabolome, which is often the byproduct of the proteome, right? The proteins work on molecules and spit out metabolites and things like that. So really going from that sort of what could be the entire biochemical pathway that our genome encodes all the way down to what really is being expressed, right? That's how we understand that journey from genotype to phenotype. Well, if we focus on the 3D genome, one of the things that it really aims to capture is the fact that the genome is pretty big. It's about 3 meters of material that has to be folded and compressed and put inside of a nucleus. And each cell has to do this. And one of the things that the 3D genome does is it captures that fundamental principle that genomes require that packaging to fit inside of a nucleus, right? So if you're thinking, if you work with, exome sequencing or ATAC or ChIP or anything like that, where you plot stuff on IGV and you see the position on the x-axis and the enrichment score or whatever on the y-axis and get those nice genomic tracks. Really powerful to create a picture of what could be happening, but it completely negates the fact that the genome isn't organized in that linear format of the genome. So that integrating the 3D genome captures that in situ information. It also has been able to uncover that this packaging is actually deliberate and hierarchical in nature. And this clustering of genomic material is something that is dynamic from cell type to cell type and from condition to condition, right? So it's a critical component that is highly mediated within the genome. And finally, that the resulting 3D structure, right, that packaging, that clustering really influences that path from genotype to phenotype, particularly with respect to gene regulation. How do we understand what causes a gene to be expressed in a particular way? So let's talk a little bit more about these three-dimensional clusters of DNA and how they're organized in the genome. So one of the things that that people who have done a lot of work in the 3D genomic space have kind of covered is there's really three main features that are captured in the 3D genome. The first one are these compartments where there are what we call A and B compartments. A compartments are typically open chromatin, accessible chromatin, actively transcribed chromatin. And then there's B compartments, which are heterochromatin, very closed chromatin that are often not actively transcribed. Now, what we found is that in the three-dimensional space, A clusters, open clusters, transcribed clusters, tend to cluster with themselves more closely than they do with B clusters, despite that, some of the closed chromatin is closer in linear space to an A compartment. We see that in the genome, the like compartments tend to cluster with each other. And then we can zoom in on those clusters, and we can see that there is sub-clustering that is happening, right? And so these much smaller structures within these compartments, we call them domains. You've maybe heard them referred to as TADs, or topologically associating domains. But again, these are just clusters of DNA that tend to interact more closely with each other than with other clusters of the genome. And typically what we find within these domains is series of linked function genes that are often mediating a biochemical pathway. And so they need some sort of synchronistic regulation to execute across that pathway. And so by clustering them together, they can kind of, the genome kind of ensures that they're going to be regulated in the same way. And then finally, we can zoom in even further and see that there are these things called loops, these chromatin loops that are often mediated by CTCF and cohesin that really act to bring gene promoters to close by to distal enhancers. So the linear distance that's separated by one of these loops could be anywhere from 10 to 100 kb, all the way up to like 2 megabase. These could be very, very things that are very, very, very part in the linear genome, but because of that looping and folding, they're actually just like a couple angstrom apart rather than megabases apart, right? And so that's really the foundational understanding of the features that can be detected through the 3D genome. And if we were to just maybe put some weight on these features, if we take two cell lines, a lymphoblast cell line, GM12878, and a breast cancer cell line, HCC1187, and we simply ask the question, how many actively transcribed genes, meaning twofold expression profile above the mean, are occurring in these features? So if I ask of the A compartments, what percentage of actively transcribed genes occur there? Well, it's about 80%, which makes sense. If they're open and actively transcribed regions, it would make sense that most of the actively transcribed genes kind of occur within those A compartments. Then if we zoom in on these domains and we ask that same question, we can see that 50 to 65% of actively transcribed genes are associated with these domains. And we can zoom in even further on these loop formations and see that 40 to 44% of actively transcribed genes just within these cell lines, right, are involved with that feature. So there's a very high probability if the gene or pathway that you're investigating, that you're interested in working with, there's a very high probability that it actually is associated with one of these features, and is at some level mediated by long-range distal interaction with regulatory elements like enhancers or silencers, things like that. And so when we think about our research and we think about the sort of approach that we take to understanding that sort of journey from genotype to phenotype, we often have very basic research questions that we want to ask, right? And so what I'm going to do is walk you through some of the technology and some of the approaches that you would use to deal with different questions. So we often have a research question, there's an approach, then we use a technology to get the data to understand that approach. Then you have the results from that data, and then you have the interpretation of that result. And so if I was to think about a gene regulation type of experiment, you would maybe start with gene expression and simply ask, what is the gene expression profile with my system, or how does it differ between two systems? And the approach you take is you go look at the transcriptome. You use RNA-seq or maybe microarray, things like that. The result here, though, is that you're detecting gene expression, and the interpretation of that result is the effect, right? There's something within that system that is making that gene be expressed. So that is the net effect of the system as a whole. Then you might ask, what factors may correlate to gene expression? Well, then you'd go look at the epigenome. This are your ChIP-seqs, your ATACs, your methyl bisulfite sequencing, stuff like that. And really, the primary result that you're going to get from these approaches is you're going to map. You're going to map where factors are on the linear genome, and you're going to correlate that factor's presence with gene expression. And so your interpretation becomes correlation, right? It's useful. It helps us identify where key factors are occurring in the genome and how they may or may not be linked to gene expression. But correlation is not causation. And so your next question might be, well, then what really causes that gene expression profile that we're looking at? So here we would introduce the 3D genome. You'd use something like Hi-C or micro-C to do that. And the result here is you're going to quantitatively link the factors of the epigenome to the expressed genes, and thereby getting a cause. You can now definitively say which factor is linked to which gene, and that gene is transcribed in X or Y, right? And thereby allowing our interpretation to be causal. So it's a really powerful tool to go from effect to cause. So if you were to just be using the transcriptome to detect gene expression and the epigenome as an ATAC or ChIP-seq today, and you'd integrate those in a multi-omic study, what it fundamentally does is it enables the description of possible mechanisms of gene transcription and really relying on that correlation to infer function. But if we bring in the three-dimensional genome, what we can really do is get measurable and comprehensive insight to the mechanisms that are driving gene expression. You no longer have to rely on correlation to get at what is the true cause of a gene being expressed. So let's take a look at this in action. We're going to go over two pretty quick case studies on how gene regulation, particularly within oncology, is really the viewpoints that the 3D genome brings that really kind of provides a very unique insight to these systems that are being studied. So the first one we're going to look at is multiple myeloma. And for those of you in the oncology space, you are very well aware that MYC is often associated with different oncogenic regimes, right? it's a cell regulator, and if there's misexpression around MYC, you're going to have cell growth that kind of just proliferates, right? And so really understanding how MYC is regulated can really provide us some unique insight, and in some cases, even new biomarkers around, around a particular phenotype. And so in this example here, we're looking at, one of our customers was looking at multiple myeloma. And they use several approaches here to interrogate what the role or what the impact of MYC misregulation could be arriving from. So first, they looked at the genome. They wanted to detect structural variants. And so if we look here, there's kind of two canonical ways that MYC is often misexpressed. The first one is through a translocation, where you have a partial gene fusion, a translocation event that takes part of a gene and puts it right next to MYC. And so if that gene is being transcribed, you'll get a partial gene product, and that the whole two will just carry on through MYC, and you'll start overexpressing MYC just because that regulated gene is now right next to MYC. Then you could have copy number variation or tandem duplication, things like that, where simply you have overexpression of MYC because you have multiple copies of MYC that are turned on. And that's probably one of the more direct ways in which we can, see overexpression just through copy number variation. The other thing that this group found is there is an epigenomic element to MYC regulation, where if you have a change in proximal enhancers, so those are transcription factors that occur very close to the MYC transcription start site, that you can actually increase transcription rates just by having more factor present really close by. And the final one, when they brought in that 3D genome, was really able to bring in this whole new class of MYC misregulation, and is now a new biomarker essentially for multiple myeloma and MYC oncogenic expression, where you have a distal enhancer that's, like I said, tens to hundreds of kilobases away, that is actually folded in and brought right next to the MYC promoter. Now, in non-cancer cases, that loop doesn't exist, but in this scenario, that loop is there, and it brings that distal enhancer right by to the MYC promoter, thereby causing more gene expression. The other thing that I did want to point out here is that the 3D genome, because it is kind of a primary sequence-based approach, the readout is off of the sequencer, you can do some really cool things. You can detect and confirm the translocation that was detected by either targeted sequencing or RNA fusion panels, things like that. And it also can confirm and detect copy number variation that you can pick up through whole genome sequencing or targeted genome sequencing. So this is actually a really, I think, a really powerful use case for bringing in the three-dimensional genome, right? If you really are trying to understand all of the potential mechanisms that could lead to an oncogenic expression regime, right? Just looking at the genome alone and just looking at the epigenome alone doesn't paint that complete picture. By bringing in the three-dimensional genome, we now have a much more complete view of screening, detecting, and describing MYC oncogenic expression. Another sort of application use case area that's really growing in our understanding of how prevalent it is something called enhancer hijacking. And this is simply where a regulatory element is now in contact with a oncogene and turns it on. And we call it hijacking because in normal cases, that oncogene wouldn't be engaged with that transcription factor at all. But in cancers, we do see that factor is now actually actively engaged with an oncogene. And so in this study here, they were looking at prostate cancer and actually found that a translocation was a key component of this enhancer hijacking. So what they did is they used the epigenome to map enhancer locations. They used the 3D genome to actually detect structural variation. They used that, again, that three-dimensional information to link an oncogene to a novel enhancer. And then they used the transcriptome to confirm oncogenic expression. So if we walk you through this example here, this is the normal chromosome 1p arm where you have DSP3 being expressed, and this is the enhancer that mediates that expression. And then you have the ARID1A gene here that is not expressed in normal conditions. Well, in some prostate cancers, there is a 1p36 inversion event, which actually puts the DSP3 enhancer right next door to ARID1A. And so through that inversion, and by bringing in the 3D genome, we can see that the DSP3 enhancer has been hijacked by the ARID gene, and that leads to oncogenic expression. So more broadly, the 3D genome, when we integrate it with other data types, really helps us in some fundamental ways in key application areas. So as I just mentioned, when we integrate it with the epigenome and the transcriptome, we build a really, really nice picture of gene expression and allows us to target the actual mechanism of gene expression. So if you're thinking about CRISPRing something out, and there are several factors nearby, and you need to know which one you should go knock out first, this allows you to do sort of forced ranking in a quantitative way about which factors are most most linked to your gene that's being expressed. And then understanding genome structure, right? Just like pure mapping of where factors are and how they're folded into the nucleus can really be highly informative in database building, things like that. Also, as we talked about in the first example, by integrating the genome, it really allows us to paint a very complete picture of the type of biomarkers that one could understand, but it also allows us to do a number of other things. So if you think about that sort of linking enhancers to promoters, that mediates gene expression, you could switch out that enhancer mark for a variant, right? Those risk variants that we know are linked to diseases, but we know occur in non-coding regions of the genome. We can use that three-dimensional linkage to help us understand that maybe this variant occurs, again, 10 to hundreds of kilobases away. We can actually link it to the promoter that it's impacting. Because oftentimes the nearest neighbor gene to these risk variants don't have any change in gene expression. So it's really hard to actually understand. We know that they're associated with a disease, but we don't know why they're associated with a disease phenotype. The 3D genome really enables that. The other really fascinating thing to me that I think we can do is do telomere to telomere haplotype phasing. Because we get linkages across the full length of the chromosome, we can say that a SNP that occurs over here in the P arm is definitively linked to this part of the molecule over here in the Q arm. And so we can build these really long phase blocks. It's kind of like doing trios without having mom and pop, except you wouldn't know which one's mom, which one's dad. You would just know that this is one haplotype, this is another. The other one that we highlighted here is SV detection. You can really start understanding a very complete map of large structural variants just by visualizing the 3D genome. It's really easy to see in the data output. And finally, sort of one of the hallmarks of the 3D genome is genome assembly, right? Once you extract your DNA and everything's all fragmented, and you sequence it and you try to stitch it back together, well, the 3D genome is a quantitative link between all the parts of the chromosome, and so you can stitch these things back together so you can scaffold almost full chromosome length. So this has been really important for bioagriculture, conservationism, evolutionary biology, things like that. But we're also understanding that it does have some implications in human disease and precision medicine and personalized genome approaches. So those are sort of the key fundamental applications in which the 3D genome can help enable. But if we zoom a little bit further back from that and more broadly say, well, what is it? What can it really do for you at a very high level? It's about bringing in causal relationships to your research. So whether you're an academic, translational research, or in the clinical or biopharma drug discovery space, the 3D genome can be highly informative. And so from a human health and disease perspective, we've seen a massive rise in customers that are using the 3D genome to study Alzheimer's and autism and other developmental and neurological diseases. Oncology, leukemia, prostate cancer, as we talked about, breast cancer, myeloma. These are all things that people are using the 3D genome today to understand. Autoimmune diseases is another big one. So various arthritises, multiple sclerosis, things like that, where we're seeing people start to bring in that 3D genome to really understand what is causing the disease phenotype expression profile that we see here. And rare diseases and adolescent cancers, right? These are cases where you kind of need to throw the whole kitchen sink at it to understand really what might be driving that particular disease. And the 3D genome is another really powerful tool to integrate because unlike FISH, where you might have to, take series of steps to detect multiple translocations, things like that, you can just do a full pass of the genome and kind of detect these bigger structural variants right away and things like that. So it's a really powerful tool in that sense. And then if you're thinking about more, clinical research or drug discovery, target ID, right? Trying to figure out where it is you need to go treat. Understanding which enhancer is linked to what promoters really brings in a whole new element to identifying what it is you might want to target. Biomarker development, the multiple myeloma cases, I think a really great example of like the world was just blind to the MYC oncogenic regime, right? They knew it was misexpressed, but they didn't know how. It didn't have those hallmark translocations. It didn't have any change in the nearby epigenome. But it turned out it was that long-range loop that was actually driving MYC expression in some cases. So, having a full picture of what could be driving that oncogenic program is I think, really important. Drug rescue, to me, drug rescue and cohort stratification are intimately linked, right? If you're developing new therapies, you want to understand why it worked in some and not in others. And if they're not differentiated on other vectors, the 3D genome is a really good place to integrate that data type to help bring in another level of stratification. All right, so now that I've talked to you about the 3D genome, hopefully there's some clarity around what it can do for you while you integrate it. We'll spend the last few minutes here just talking about how you can get the right 3D genomics solution for you. So maybe a little biased, but we do think going through the Dovetail solutions is a really good approach to meeting your 3D genomics needs. We have over 8 years of experience working in this 3D genomics world, and we're really excited that it's starting to really spill out into much broader application areas than just mapping the tags and the loops, and really starting to add that functional component to people's research. And so we have kits that are available. Now, some people might like to homebrew and make their own master mixes. There's some really key components of purchasing pre-formulated kits, right? The validated reagent quality, reproducibility, and another big one is customer support. If you're making your own chemistry and it doesn't work, you really don't have a neighbor or anybody to call and be like, well, maybe why didn't this work the way that I expected to? We're here for you to phone a friend and help you understand, how to get the most out of your experiment. Services, we know not everybody either has the time or bandwidth to onboard new technologies and things like that. Perfect. Send us your sample, we'll send you the result. We have really great end-to-end solutions that are very flexible, and we have dedicated project managers that will really help guide both you and the project through to completion and making sure that you get what you need. And one of the other things that we really strive to do with both our kits and our services is value. We really want to ensure that the value that we see in the products is the value that you're getting out of the data. And so one of the things that we think about is increased savings. not just in terms of the actual component of the process that we sell, that kit or service. We're thinking like, how does this impact your sample requirements? How is this going to impact your overall sequence depth, right? How is it going to impact your overall reagent use, both within and outside of the chemistry? So we really want it to kind of make sure that you're getting the most out of what you're spending. The next one is improved accuracy. Of all the approaches to the 3D genome, what we do with the Dovetail assays has the highest accuracy and least biased approach to interrogating the 3D genome. And with that, we also have the highest sensitivity. We have the highest signal to noise of any other approach to really ask questions about the three-dimensional genome. So we have a couple approaches for you to take when interrogating the 3D genome. The first one is global discovery. If you've never used the 3D genome before, or you don't really know what factors are important, if you even don't know really, what feature it is you should be looking at, well, you can do the whole genome. You can get the whole genome three-dimensional structure. You can capture all of the features, those compartments, those paths, those loops, and it's very unbiased, right? You're going to get a complete picture of everything that's in that three-dimensional genome. The other approach that we have are target enrichment approaches. So this can significantly reduce your sequencing costs. It can vastly improve your testing power because now your reads are now concentrated on fewer tests. And it can increase your sample size. So with global discovery, your budget may only allow for a small handful of samples to go through to sequence the full genome. But if you have a targeted approach, maybe that sample size goes to 20 or 30, and you can now really add power to that biomarker discovery, or to really understand how broadly a gene expression mechanism occurs across a phenotype. So the products that we really have are really focused on driving that three-dimensional genome discovery. The first one is our micro-seq kit. Now, this is the best-in-class approach to 3D genomics that there is. It's the highest signal to noise. It's really sensitive, and it doesn't really have any base biases. So you're going to get highly accurate feature calls within this. And it builds a complete picture of the 3D genome. Our next approach is something that we call hijacking, again, using MNAs to fragment the chromatin. But it takes the benefits that are the hallmarks of micro-seq and combines them with ChIP-seq. So once we have our sample fixed and ready to go through that 3D genome, we do a chromatin immunoprecipitation, pull down on a factor of interest. And so now we get the protein binding site, and we get the three-dimensional contacts associated with that binding site. So we get a factor-specific interactome view of the 3D genome. And finally, we have pan-promoter panels. So if you have a ton of factors, like you've done a lot of ChIP-seq against a lot of different antibodies, and you really want to understand, well, which ones are the ones that are really driving gene expression, right? You don't need a factor-centric view. You need a gene-centric view to understand what genes are intimately linked with what factors. And so again, the pan-promoter approaches leverage that, although the benefits of micro-seq, and then we combine hybrid capture with that. So we target gene promoters in that three-dimensional genome, and that allows us to save up to like 90% on sequencing, depending on the approach that you're doing to interrogate your three-dimensional genome. One of the other, maybe not so scientific, but I think a fun component of the Dovetail solutions here at Cantata are really the events that we have. We do very frequent webinars that span the range of very broad, high level, kind of like what we have here today, all the way down to the nitty gritty about exactly what aligner do you want to use? Exactly how would you visualize some of these data? How would you do annotation? Or simply how to pipette the kits, right? And tips and tricks, things like that. And so one of those sort of more technical webinars is coming up here, as Jesse pointed out at the start of the talk. There'll be two, one on August 9th and one on August 15th, where Miriam, who's absolutely a fantastic scientist, will be there to answer any and all of your questions around how to set up a 3D genomics experiment. It'll be split 50-50 between, she'll have a kind of overview of what that process is and what it looks like, but really she's there to answer your questions. And so come there with all the questions that you have, and she'll be a great resource for you. The next one that I want to highlight here is our DT Epi Summit. We do this annually, and we're really excited. We have a great speaker lineup. This is kind of like a user group meeting, but it's really a place for our customers that are using 3D genomics today to really talk about their cutting edge research to other folks who may be a little bit on the outside looking in and maybe wanting to understand a little bit more about specifically how these data types will answer their research questions. The other really cool thing about DT Epi is we often use it as a platform to announce big updates and big advancements that are associated with the Dovetail product line. And just a little teaser, there's a lot coming down the pike for this September. So we would love to have you there. And at this point, I think that's it for me. So I'm going to pause now and start going through questions. If you have questions, please put them into the Q&A, and I'll answer them from there. So what type of data do I need to do 3D genomics? Is it okay with genome and Hi-C? Yeah, so Hi-C is kind of like the background technology. I would say the historical technology. As of about 3 years ago, micro-C has rapidly started to displace Hi-C in the research world for a couple of reasons. One being resolution. Micro-C, you can get really high resolution mapping of the chromatin interactions. But the other big one is bias. Hi-C data is incredibly biased towards restriction site, because that's what you use to fragment the genome. We have a lot of webinars and recordings on this. I really encourage you to check out our website. But one of the big implications of using restricted enzymes for loop detection and TAD boundary detection is you can only call features where you have data. And what we often see is that TAD boundaries and loop anchors are shifted by a handful of bins, not because of any biological signal, but simply because there's more restriction motifs, 10 kb away from the actual binding site. And so the net result is you bias your data to something that might not actually be accurate. So really, it's not just us. We've seen the world kind of move into micro-C as kind of being the clear-cut winner. There was a really great conference a couple of months ago. It was a Keystone Symposium, where it was very clear, that micro-C was just being used a lot more broadly than Hi-C was. So yeah, thanks for that question. The next one: can the 3D genome be useful in understanding bacterial genomics? I am a microbial bacterial person as a background. There are some applications for it, so you can use it to maybe do metagenomic genome assembly, where you use the linkage within a cell to go back and rescue contigs and bring them together, kind of like you were doing, it's like the metagenomic binning, except instead of using tetranucleotide frequency and coverage as your vectors to build your clusters, you could use that 3D contact to do that. There are a lot of caveats in it, just as there are a lot of caveats with the sort of metagenomic binning process to generate genomes. The other place in which there's utility is mapping plasmids to the genome of origin, right? Because as soon as you extract your DNA, that plasmid is no longer associated with the cell that it came from. And so if you're looking at, antibiotic resistance and things like that, those often occur on plasmids. So understanding what species or strain that plasmid came from can also be useful. I would still say it's fairly early days in that realm, if I'm being honest. with you. Doesn't mean you can't do it. Doesn't mean that it's not doable. It would just probably take some thinking on your part about really, how would I use this information to help me understand. Okay. So I got a question here. How exactly are 3D features preserved during sample processing, whole genome discovery, or targeted? So the targeted approach happens after. It happens after the 3D preservation. We use formaldehyde and DSP to fix. We find that those two approaches really preserve that 3D structure very, very, very well, and allows us to interrogate that. And so the first step is always going to be to fix your sample. Then it's going to be to fragment, and then if you're doing whole genome, just carry on the micro-seq protocol. If you're doing targeted approaches, then you'll do the chromatin IP and then do the rest of the micro-seq kind of workflow. Or for the pan promoter, you would just do full-on micro-seq, and then with your final library, that's when you tack on the hybrid capture. Cool, that was a fun question. Minimal cell numbers for high chip promoter kits. Yeah, so for our targeted approaches, for the pan promoter panel, it's 1 million cells. That's what you need to carry out the work. Same thing for micro-seq, 1 million. For a high chip, you're going to want somewhere between 5 and 10 million, just because it's a little lossy, right? When you pull down on the antibody, you're losing a lot of DNA, right? And then you do proximity ligation, and we biotenylate in that process, and we pull down again when we do the library conversion, because we only want to take those ligated products into the final library. So you lose a little bit more DNA there. And so really the name of the game with high chip is balancing your input to having a sequenceable library that's not full of PCR duplicates. And we found that somewhere between 5 to 10 million is a good place to do that for applications like loop calling. Okay. We got a question about promoter capture on micro-seq. The answer is yes. That is our pan promoter. I love when people ask me questions that I already have the answer to. Love that. Yeah, so our pan promoter panel is the thing that does that. Jesse, did you need to hop in for anything?
Yeah, no, I was just going to say, if I can insert in a moment. Thank you so much for such an awesome presentation, Cory. It was so good. I'm just going to pop up a poll for people while they're listening to the answers of the questions. If you guys wouldn't mind, it's so helpful for us to understand what kind of research you're doing and what your interests are. Very helpful. So I'm going to pop up that poll. Carry on. That's all I wanted to let you know.
Cool. Thank you. I really like this next question. So the targeted 3D approaches like the pan promoter panel be used to detect breakpoints in the genome for like some sort of structural variant, assuming that they give rise to expressed fusion transcripts at the RNA-seq level. Typically, you're getting fusion at 2 exons, but at the DNA level, the breakpoint is intronic. Love this question. The answer is, yeah, absolutely, you can do that. There's a couple different ways. For the targeted approaches, you would probably want to take a look at what we have and see if it spans your breakpoint. If not, it would be sort of like we would work with you to maybe build a panel that would meet your needs there. But absolutely, you can capture that. The other way to do it, it's not targeted, is with relatively shallow sequencing with a micro-seq style library. You'll be able to span those breakpoints really easily, because instead of like long read sequencing where you only need one, or where you need to have multiple reads that span that breakpoint, we have lots of reads that stack, plus or minus 5 KB of a breakpoint, all the way from the breakpoint all the way back. And those contacts, that 3-dimensional information, actually capture the fact that this molecule is jumping a breakpoint. And so we have a lot of evidence of where these variants are. And exactly, you don't have to rely on exons. You can get into the you get to the base level breakpoint, which is really, really cool. And I think So one of the really nice parts about the 3D genome, it combines that primary DNA sequencing, particularly the way that we do it with the Dovetail products, is if you think about the molecule on a single end read, it's just whole genome shotgun data. There's nothing special to it. When you combine the proximity ligation or the second read in the mapping, now it becomes informative of the 3D genome. And so all the stuff that you can do with whole genome sequencing, you can do with the 3D genome, particularly with the way that we do it, because we don't have any bias towards particular DNA motifs. So yeah, super cool question. I would really encourage you to reach out and talk with us about that. Oh man, questions keep rolling in. I like this. Okay, mentioned that the 3D genome is causal and that epigenome is correlative. Let's see. Hold on. I think the epigenomic factors are pioneers to determine 3D genome information, and the 3D genome can give additional effect of the localization of the epigenomic factor. If this was generally the situation, then I think the epigenome is actually causal and the 3D genome is correlative, the factor. How do I think about this? I think you bring up a good point in that, you know, for proximal enhancers and stuff like that, there is a case to be made that they're probably causal at some level. The 3D genome does, you know, commingle with the epigenome quite frequently, right? If you think about, you know, a zinc finger protein needs to bind and a cohesin ring needs to be recruited to form a loop, right? So that's in essence a epigenetic factor that leads to a 3D component. But one of the things that you often cannot do with the 3D or without the 3D genome is quantitatively link those distal factors to the promoters. Right? It's just there's no good way to do it. You have to do it through correlation. And when you have a space of like 2 megabases that you're trying to understand the regulatory dynamics within that space, there are tons of factors that occur. Lots of chromosome opening and closing, lots of histone modifications, right? Lots of cis regulatory elements that are binding. And without the 3D genome, you can't definitively say one way or the other, which one of those is the key driver, unless you want to do tons and tons and tons of knockouts within that space, right? So I see your point. I think it's a little bit of both there, where one informs the other in some cases. But when you're really trying to paint a full picture of gene regulation, I think the broader 3D genomics community is kind of centralized around like, this is the way by which you quantitatively link things to promoters. So I don't necessarily disagree with that comment, but I also think there's a broader picture to be had there. But yeah, that was a very cool comment. Thanks for that. What is the starting material for 3D genomic cells or extracted DNA? So you need cells or tissue. You need the intact cell to do this work on, because as soon as you extract, you know, one of the first steps in DNA extraction, right, is removing all the proteins, and we need to fix those proteins together to make that to keep that 3-dimensional structure intact. So we do start with whole cells, and just, you know, if you have extracted DNA, well, it's extracted. That 3D information has been lost and can't really be recovered. What is the best approach to study long-range multi-megabase loops formed outside of TADs or compartments? Ooh, I like this. There was a really great talk at that Keystone Symposium back in March that actually used micro-C on fly embryos to really look at these long range loops. Like we're talking like 4 megabases apart, and there's this one punctate spot that sits way above any of the other topology features. It was super powerful, and they used microC to do that. So that would be our suggestion there. You know, once you're looking at features that are that far off the axis, you should be able to pick them up pretty readily, without having to like do like ultra deep sequencing or anything like that. But yeah, I mean, we often see those too when I'm just like scanning the results. You can see some of the handful of these like really long loops. The other interesting one too is you can start doing that in the trans space. So maybe a couple chromosomes share a super enhancer hub and things like that, and you can actually go back and map out which promoters are all from different chromosomes are sharing the same super enhancer cluster, which is pretty cool. Cool. Well, that takes us to the top of the hour. Thank you all for your questions. It's really fun to have an engaged audience. So I think I'll stop talking now and hand it back over to Jesse.
Yeah, thank you so much, Cory, and thanks for answering all those questions. I wanted to remind everyone that I'll be sending the recording out and the slides. So you guys, if you missed anything or you want to review the information, you'll be able to do that. And I'll be sending that out in a few days. Thanks so much, everyone, for being here. We really appreciate it. And it's great to connect with you all. And we look forward to connecting with you again. Hope you can join us for the next webinar, Ask the Experts, on August 9th and the 15th. All right, take care, everyone. Bye for now.